So tonight we'll hear from another of Rockefeller's truly illustrious faculty, Dr. Jim Hudspeth, who's a world leader in using physics to understand the complex operations of sensory processing in the nervous system. As the university's FM Kirby Professor and a Howard Hughes Medical Institute investigator, Jim focuses on the sense of hearing, which plays a huge role in our lives, but depends on a compact arrangement of receptor cells that reside in the internal ear. When Jim began conducting his research in the late 1960s and early 1970s, very little was known about hearing beyond basic anatomy and physiology. His work has led the way in establishing how hair cells convert the energy of sound waves into electrical signals that are relayed into the brain for interpretation. He has dedicated his career to understanding how hair cells enable these remarkable feats. Jim's biography says he was born and raised on the outskirts of Houston, Texas, near a large natural park that was populated by amphibians, reptiles, birds, and small to medium-sized mammals, of which about 200 could be found in Jim's home at any one time, much to the joy of his parents. His interest in fundamental biology grew during summers spent assisting a neurophysiologist at the Baylor College of Medicine, and he then went on to get his undergraduate degree from Harvard College and entered the doctoral program at Harvard University. There, his thesis advisors were Torsten Wiesel and David Hubel, who are remarkable neuroscientists whose research on the visual system were recognized with the Nobel Prize in 1981. And Torsten Wiesel, as you may know, joined Rockefeller as a professor in 1983 and served as the university's president from 1991 to 1998. So Jim also received an MD from Harvard Medical School and followed this with postdoctoral work at the Karolinska Hospital in Stockholm. He next served on the faculty of the California Institute of Technology, where he and his colleagues started their pioneering work about how hair cells can transduce mechanical stimuli into electrical signals. He moved on to the University of California at San Francisco, began to devote more attention to the biophysical basis of hair cell function, and then returned home to Texas to work at the University of Texas Southwest Medical Center in Dallas, where he became an investigator of the Howard Hughes Medical Institute. 2 years later, he moved to Rockefeller, where he established the Laboratory of Sensory Neuroscience and has been here for the last 30 years. The Hudspeth Lab uses biophysics, electrophysiology, biochemistry, genetics, molecular biology, and developmental biology. And in addition, he and his associates assimilate their findings into mathematical models of hair cell function. Jim's many contributions to the field have been recognized by numerous honors, including the Louisa Gross Horvitz Prize from Columbia and the Kavli Prize in Neuroscience. He's a member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences. Jim's work exemplifies the principle that great basic science is not only intrinsically beautiful and informative, but also lays the groundwork for improving the lives of millions. His foundational work provided the basic understanding of hearing that has allowed the development of cochlear implants, which has brought hearing to nearly 1 million people around the world over the last decade. More recently, Jim and his colleagues have begun exploring therapeutic approaches that have the potential to reverse the loss of hair cells due to injury and aging, which impairs hearing in about 40 million people in the United States alone. That number is certain to increase as our population continues to skew toward older age groups. Jim's ambitious goal is to identify safe and potent therapies to replace lost hair cells by regenerating new ones. These initiatives have emerged through the Hudspeth Lab vital insights into the development, structure, and operation of hair cells. As we'll learn tonight, Jim and his colleagues have made some incredible findings with implications for the treatment of hearing loss and other health conditions for which no truly effective treatments are available. So now, please join me in welcoming Jim to the stage.
Thank you, Rick. It's an honor to welcome the friends and supporters of Rockefeller University. And as you've heard, I work on hearing, and I'll be tonight discussing a project that began as an excursion into helping with the repair of the internal ear, but it's spread more widely and opened avenues to possible therapies in a number of fields. So I want to begin with the hearing part by talking a bit about what is going on right now in your ears. So this is a spectrum of human speech. What it's showing is, along this axis, you can see time. It's about 4 seconds of human speech, of my speech. And along the other axis is frequency, that is tone. So these are low frequencies down here. These are high frequencies up here. And what you can see is the complexity of speech. This is what we take for granted. We're hearing signals like this all the time. You're hearing them right now. But look at how complex they are. They're low frequency stacks of frequencies, harmonic frequencies. They're high frequency swishy things, and so on. And this particular speech is, in fact, what I just said at the outset. It is an honor to welcome friends and supporters of Rockefeller University. So you can see, for example, let's take any old word. The word friends begins with this f with a high frequency, in s, and supporters of Rockefeller University. It's remarkable, right, when you think about it. In this talk, I will use something like 20 or 30,000 speech sounds. During a day, you may hear close to 1 million, and yet you're not surprised at the end of the day that you've understood everything you've heard. So your ear and your brain have to be continuously capturing this complex stream of things and somehow breaking it down into its constituents and figuring out what has been said. Now, the apparatus for doing this is, of course, the internal ear. Sound consists of vibrations in the air outside that impinge upon the eardrum and set it into oscillatory motion. And that motion is conveyed through these 3 little bones, the last of which acts as a tiny piston, increasing the pressure on the liquid inside the coiled cochlea, the organ of hearing. And the cochlea is also associated with the vestibular apparatus. This is the apparatus that accounts for our ability to balance our sense of equilibrium. Here is a classical etching from 1884 showing this beautiful structure, showing its spiral nature, and showing the 20,000 nerve fibers that carry information from this into the brain. Now, we all know that a prism can break down different frequencies of light, that is, different colors, into their constituents. So white light coming in here is fractured into all these different colors. And in a similar way, the cochlea breaks down complex sounds into their constituents. So relatively low frequencies occur here near the upper end of the cochlea. High frequencies occur down here towards the base of the cochlea, and intervening frequencies occur partway in between. So there's a continuous map along the cochlea from the lowest to the highest frequencies, always in a neat order like a piano, though of course the cochlea extends to lower frequencies than a piano can play and higher frequencies than a piano can encompass. Along the length of the cochlea, the cells that do the detection of sound are called hair cells. And the name is something of a misnomer. It has nothing to do with the kind of hair that I have less and less of. In fact, it was given that name by classic microscopists because there's this so-called hair bundle, these tiny little processes, little feelers sticking out of the top surface of the cell. And as I'll show you in a moment, that hair bundle is the thing that actually responds to sound, or in the vestibular apparatus, to accelerations, to motions. This is otherwise a normal cell. It has a cylindrical cell body, it has a nucleus down here, and its other feature are what are called synapses. These are the sites at which chemical information is passed from the cell to nerve fibers that then carry the information into the brain and tell the brain, yes, I just heard such and such a frequency of sound. Now, the hair bundle, shown here in a living cell, is an elastic structure. If I press upon this living hair bundle with a little glass rod here, you see that the bundle can move one way or the other. And this motion, in fact, recapitulates what's going on in your ears right now. Each of the hair bundles in your ear that's responding to sound is vibrating back and forth in response not to a glass rod, but to the pressure from the ear into the ear from sound. So the energy that you're hearing is actually transduced or converted into back and forth vibration of the hair cell, and the back and forth vibration of the hair bundle then sets up electrical signals that the brain can interpret. There's an elegant pattern of these. Here we see something on the order of 100 of these hair bundles sticking out of the top of successive hair cells. Our cochleas each have something like 15 or 16,000 hair cells. They occur in rows, typically of about 4. These are also hair bundles down here. Now, 15,000 sounds like a lot, but of course, that's a relatively small number of cells. It's about as many cells as you would get in a hangnail or maybe a fleck of dandruff. And all of your hearing depends on just those cells. If you lose those cells, you are deaf. And it's terribly important that we sustain those cells, and we're now, of course, trying to replace them when they are damaged. Now, it follows from what I've told you that different cells at different positions are responsive to different frequencies. So if these cells, say, happen to be sensitive to middle C, the cells about this far away would be sensitive to C sharp, and so on for all the tones as you go on up. So you can think of this as an array of little detectors, each of them responding to specific frequencies along the whole 3 spirals of the cochlea. Hair cells don't occur in a vacuum, but each hair cell is surrounded by what are called supporting cells. And the supporting cells are essential for the maintenance of the hair cell. They provide metabolic factors, they mop up some problematic chemicals that the hair cells produce. And so it's essential that they be there for the hair cells to be healthy and happy. Unfortunately, hair cells become damaged. So here's the hair bundle of a normal hair cell with the neat pattern that I showed you before. This is in a human ear. And here's another human ear in which damage has occurred. And you can see that the hair bundle has become highly irregular. This cell will proceed to die, and the cell is not replaced by cell division. There are no mitoses or cell divisions in this system. So once that cell is gone, it's gone for good. And once a series of cells have gone, the person will have lost a particular range of frequencies that will not be replaced. And this is why, in fact, our ability to understand tends to deteriorate as we lose hair cells. We lose sensitivity to successive frequencies, and it becomes harder and harder to interpret the very complicated sounds that I showed you in the initial slide. Hair cells can be lost by a number of mechanisms. Acoustic trauma, which means very loud sounds, whether from an industrial or perhaps military circumstance, from playing music too loud or whatever. There are various infections, such as meningitis, that can devastate these cells. There are pharmaceuticals that damage them, and there are 2 of particular importance. One is cisplatin, which is the major chemotherapeutic for ovarian and testicular cancer. And sometimes that has to be used. It's a life-saving measure, but it can cause partial or complete deafness as a consequence. The other set of compounds are the aminoglycosides. These are things like gentamicin, tobramycin, neomycin, and whatnot. They are used sometimes for severe infections, say endocarditis, infection of the heart. Again, they have to be used if it's a question of life or death, but they have as a consequence devastation of hearing. And finally, just plain aging obviously whittles away hair cells, and we lose hair cells in an environment such as New York City from a combination of aging, but also acoustic trauma. Every time you hear those terrible noises in the subway or the like, you may be suffering some lasting damage. Now, in non-mammals, this is not a problem. In non-mammals, these supporting cells that survive can undergo proliferation or multiplication, and some of them then convert into hair cells, and they're back to normal. So a chicken or the like can be subjected to deafening noises or other conditions and get its hearing back almost entirely within a matter of a few months. In the case of mammals, this does not occur. and we don't yet understand why. But in mammals, what happens is the supporting cells do not proliferate. Some of them do undergo what's called transdifferentiation, meaning they simply turn into hair cells. But the problem is, without the surrounding supporting cells, these cells are undernourished, they're not properly supported, they tend to die and leave a scar, and that is permanent damage. So in looking at the difference between these two situations, the striking feature is this proliferation. In one case, there's none. In the other case, it's quite prominent. And that led us to want to try to recapitulate what happens in non-mammals. That is, to get mammalian supporting cells to undergo division in the hope that we can then create new hair cells. So we undertook to do this in the mouse's ear, and we use an organ there called the utricle. Here's a mouse's ear with the spiraling cochlea in an embryonic animal. The utricle is another organ that has the advantage that it's flat, it's large, it has about 2,000 hair cells. So this particular label, the pink one, shows all the hair cells in a normal utricle. This is an organ of our vestibular apparatus, and it's what enables us to sense motion in the plane in which we walk. So if I close my eyes, I won't run into Rick, I can move over here, I can move back, I can come back to where I started. And it's because I'm measuring the acceleration in each of those directions with my utricle. The brain processes that information and says, okay, here's where you are. The other organ, the saccule, does the same thing in the up and down direction. This is what you feel when you're in an airplane, when you're seasick, when you're in an elevator, or whatnot. But this organ is very convenient because it's easy to dissect, and it's very easy to assess the number of hair cells and other cells. That's much harder in this complicated cochlear structure. So we began our study by asking what genes turn off when the ear can no longer make new supporting cells. So in a mouse, around the time of birth, which is defined as P0, postnatal day 0, there are still a lot of cells being made. These cells are labeled with a compound called EDU. It doesn't matter what it is. But every white dot here in the center represents a newly formed supporting cell. And the number is on the order of 20,000 per millimeter squared. Quite a lot. But just a week or so later, P9, the number has fallen to just a handful, and it subsequently falls to none at all. So the capacity to make these cells simply disappears in the first weeks of life. As you can see, it disappears very briskly around the day of birth. So we ask what genes change at that time that might account for this loss. The hope being that if we could understand what genes are turned off, we might be able to turn them back on and restore this capacity to proliferate. We found, among other things, genes that are called SOX-C. It doesn't matter what they are. Their names are SOX4 and SOX11. But these are what are called transcription factors. They're genes that switch other genes on and off, and they send information about what fates different cells should adopt. And realizing that those cells might be involved, we made mice in which those genes were switched off permanently. So here's a normal animal, and soon after birth, we see this beautiful pattern of hair cells forming, just as the sort that I showed you earlier. But in an animal in which those genes are switched off, you get a complete lack of that orderly arrangement of cells. So this is a strong hint that these particular genes are involved in laying out the supporting cells and the hair cells in the cochlea. And we wondered then what impact they might have. And it's well known that some downstream genes that those genes affect include a particular pathway. And I won't talk about details of the pathway, I've simplified it here, but the pathway consists of these proteins here in the center, and I've just called them A, B, and C for the sake of argument. But here's what this pathway does. If you're a normal cell in a sheet of cells, let's say your skin, you are ordinarily surrounded by other cells on all sides, and everything is good. You're happy with that. So these cells feel their junctions, their contacts with neighboring cells in each direction. And when those contacts are present, that sends chemical signals to these proteins, and finally to this protein, and this protein is destroyed. It gets chewed up, it gets bound. It has no influence. This is sort of a silly way to send information, but this is the way the body does it. If, on the other hand, there's a wound, let's suppose your skin is cut, now the cells along the edge of the cut don't have neighbors anymore. They've been isolated. And in that circumstance, what happens is there are chemical changes, first in this protein, then in this one, then in this one. And this protein, when it's changed, is no longer degraded. Instead, it migrates into the nucleus, and it turns on genes. That's what this symbol means. And the genes that it turns on are genes that are involved in making new cells, in replication, in proliferation, in cell division. So this is the mechanism by which ordinarily wound healing occurs. If there is a cut, the cells on the sides of the cut proliferate, they grow, and they patch up and fill the wound. So knowing that this pathway is involved in the suppression of supporting cell production, we reasoned that it might be possible to intercept this message. If we were able to intercept this message, say with some drug, placed at a critical position, even normal cells, like supporting cells that have junctions with their neighbors, they would send the normal chemical messages, but we would stop the message from getting to protein C. Protein C wouldn't know any better. It would go into the nucleus and trigger proliferation. So this is a mechanism by which we can seduce a cell that's in normal circumstances to undergo another division. And that's what we want to do to make new supporting cells, and therefore to make new hair cells in the ear. So how do you do that? At the outset of this adventure, I knew nothing about drugs except what you see here. Every morning, my wife hands me something like this. She might be poisoning me, for all I know. If so, it's taking a long time. I never thought about what's in these things, and most importantly, how it gets there. And we then found ourselves wondering, how can we find a compound that might do what we want, to intercept those signals and thereby create new supporting cells? And the way that's done is schematized here. One can make little dishes, little plastic dishes. I had one here somewhere. And in these dishes, you have lots and lots and lots of tiny wells, tiny little chambers. This has 96. You can get them with up to 384 separate little wells. So you put cells in each and every one of those wells. You then can add a distinct chemical to each and every one of them. So one can buy not just thousands, even millions of different chemicals that have been synthesized and are commercially available. And one can then place those chemicals in the wells and ask, does a particular chemical, any one of these thousands of chemicals, affect the process you care about? We benefited enormously in doing this from the support of something called the Robertson Therapeutic Development Fund on this campus. This provides support for exactly this kind of project. If one has, in our case, a sort of flimsy idea of what might be possible, they will nonetheless consider it and perhaps support it. And that support is necessary to carry out a scale, a project of the scale involved. We looked at 140,000 compounds, 140,000. And from that, we found 6 that had the right sort of properties. 2 of them were good at causing proliferation of the cells in the ear. 1 of them was not toxic. So 1 hit out of 140,000. But that would not have been possible without this support. Now, when I say we did this, I don't mean that we did it. The robot did it. Pipetting 140,000 different drugs into 140,000 little wells would be beyond maddening. This robot, in something called the Fisher Drug Discovery Resource Center, did all of the work. And this highlights the importance of what are called resource centers on this campus. There are 15 of these centers, and each of them is a specialized facility available to all the faculty, all the students, and all the postdocs for performing certain specific sorts of tasks. And they are enormously effective. They're effective for really 2 reasons. One is that it costs a lot of money to get a fancy robot like this, and the resources are much better spent buying one high-quality machine for the whole campus and then sharing it, than having dozens of cheaper machines in different laboratories running in parallel with each other. The other important issue is each of these resource centers has really talented personnel who are highly trained, most of them are doctors, who know how to use the apparatus. I have no idea how to talk to this or any other robot, but the point is we simply give them the project, we give them the cells, we tell them what we want done, and then the people involved can program the robot which takes over and does the hard work. It also measures the responses in all of these 140,000 little wells and tells us which ones look like promising hits. I should mention Evie Lipper, who's here tonight, has been instrumental in setting up the cryo-electron microscopy resource center, one of those 15, which has been an enormous addition on this campus that lets people who wish to do analysis of complex protein structures that were not accessible before by any other technique, including X-ray crystallography. So this is what we got. This is the 1 in 140,000. And it looks like every other chemical. It looks inscrutable. It has different kinds of atoms. So there are carbon atoms at every corner that isn't otherwise labeled. There are 4 nitrogens labeled with an N. There's 1 sulfur. There's 1 oxygen. And there's this funny sort of an arrangement. All we knew about this is that it works. We had no idea what it was doing. We didn't know exactly how it was reacting, but we knew that it had the right sort of quality. Here's, in fact, the result. I showed you before that after a few weeks, you get very few new cells, these white ones, in the center of a utricle. If we apply this compound, we get hundreds, even thousands. So it's enormously effective. That's as far as we got. The next question is, what do we do with this molecule? And is the molecule the best it could be? Well, given all of these different atoms, one can ask, how might they be rearranged? Could we tweak it, by adding a little embellishment here or a little embellishment there to make it better? That is a very complicated process. This doesn't look so formidable, but if you imagine just having, say, 2 choices at each of these positions, that would have about 16 million possible variations. We don't have time in many lives to look at 16 million of these, but we have help. And we have help from something called the Sanders Therapeutic Discovery Institute, TDI, which is now on our campus in the Bronx Laboratory. And this is a remarkable resource. It's tri-institutional, so it's supported by Rockefeller and also by Memorial Sloan Kettering and by Cornell. It has very talented people, many of whom worked previously in the pharmaceutical industry, who have an intuition and a lot of experience in how to think about ways of modifying the molecule that are most likely to be fruitful. Nobody can do 16 million of them, but they in fact did 250 modifications. And the numbers are shown here. So 180 changes here, 96 here, 71 here, and so on. And these were all somewhat intuitive, but also quite informed choices. And this is a lot of work. All of these had to be chemically synthesized, so the total bill for doing this sort of thing is certainly in the millions of dollars. It's a huge enterprise. But it was extremely effective, and again, it's an enormous resource that we have available because with the support from Rockefeller and the other institutions, this is essentially free. The facilities and the remarkable expertise are given to any project that's considered worthwhile. So having shown that we can cause replication in the ear, and we have more and more examples of that, we then asked, does it work elsewhere? So we looked at what are called retinal organoids. These are cells that are human embryonic stem cells grown in a dish that are convinced that they are going to become retina. In other words, they're changed into cells of the retina of the eye, even though they're still growing in a dish. And they are a very good system for asking questions that will also apply in the eye itself. So when we look at this system, we see when these organs are subjected to just control conditions There's a very small amount of incorporation of this drug, EDU, which says, yes, these cells are replicating. They're undergoing division. I've circled one cell here that's turned red. Here's another red cell. So a couple of cells have turned red as a result of spontaneous replication. But if we put on our initial drug, we see instead of 2, dozens. All of these red, orange, yellow cells are in fact cells that have undergone reproduction, and I've circled them in half of this diagram. So there are 18 of them here, and there are a similar number on the left-hand side of the diagram. You can see a lot more red, orange, and yellow cells. So these compounds cause an enormous amount of proliferation in the retina as they do in the ear. And in fact, if we use our latest drug, a most effective one, you can see an uncountably large number. I didn't have enough little circles to label all the red, orange, and yellow cells. It looks like candy, but they're replicating like mad. So a suitable amount of this compound will produce enormous numbers of new cells. And these cells could go on to reproduce and to replace damaged cells in the retina in circumstances such as retinal degeneration and other similar conditions. Now, does this work in an intact animal? So here's an animal that's not treated with anything, and all of these blue nuclei are the nuclei of photoreceptor cells that capture light. These other nuclei here and here are cells that process the information and cells that send it into the brain. And you'll see there are no cells labeled with this compound EDU. But if we treat with these compounds, we find now 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 cells have taken it up, and they've not only reproduced, but they've begun to migrate to other layers of the retina, indicating that they're making new nerve cells. So we are now doing experiments in collaboration with 2 former postdocs at USC. We're doing experiments asking whether a damaged retina that's been exposed to too bright a light, to laser pulses or whatever, can actually replace photoreceptors or other cells. But it's very heartening that even in an undamaged retina, we get some replication of cells. We think a damaged retina will cause far more. The last thing that we've done here is to work on the heart. Of course, cardiac cells, heart muscle cells don't regenerate. That's the reason, in fact, that heart attacks are so devastating. But while we had this compound, we decided to check in the heart and see what happened. So we made small lesions. We damaged the hearts of mice with a little bit of freezing, and then asked whether any of the muscle cells there also would undergo proliferation, and the answer is yes. So a cardiac cell, a muscle cell cut across, has this red label on it, which is a muscle protein. It has often a green sort of glow around it, which is extracellular material. These are little arteries running nearby. And the cells that have undergone division or undergoing it, again, incorporate EDU, this white compound, into the nucleus. So we have 1, 2, 3, 4, 5 examples of cardiac cells that are undergoing division, and we counted thousands and thousands of more. So what we're trying to do now is to scale this up in animals that have had a real heart attack. So for example, to take mice or rats, tie off part of the coronary artery to mimic a heart attack, and then ask, can injection of this compound into the animal cause a significant number of new cells to form, and therefore cause some healing of the heart attack lesion? So that's where we are today. We've gone some distance from the original work on hearing to more and more other systems, and there are yet additional systems in which these same compounds are applicable. We're collaborating with people at Weill Cornell who are working on adipogenesis, the formation of fat cells. That's affected by this. Interestingly, the progression of prostate cancer is affected by the compounds. Prostate cancer typically undergoes a transition to a very dangerous metastatic form that's called neuroepithelial cancer. And this set of drugs somehow blocks that transition, so it might have therapeutic implications. It affects the growth of embryonic stem cells. That's being used, again, by people at Weill Cornell. There are also commercial entities that are interested in it. And most recently, we've learned that it has effects on muscular dystrophy, totally unanticipated. but these compounds can facilitate processes that keep experimental animals alive longer in the presence of muscular dystrophy. So again, we have hopes that this might turn into a human therapeutic. Other published studies that we have nothing to do with, these compounds can now be bought. So other people have found that they cause a reduction of cell death in lupus. They control an infection with Clostridioides, which is a bad pathogenic bacterium that's often found in cancer patients. It can cause a lot of morbidity. They are even being used in dentistry to look at mineralization of the bone around teeth and to improve that. They protect inner ear cells from cisplatin. Remember, I mentioned that before is the chemotherapeutic in ovarian and testicular cancer. This may have a protective effect, mitigating the damage that that causes. And they also seem to protect neurons, brain cells, after a stroke. So our hope is, at the very least, these compounds are a very valuable tool. They are cheap. I worked out this morning, they cost more than cinnamon, but less than saffron. And so there are, as has already been mentioned at the outset, there are 40 million Americans who have significant hearing loss. That's a lot of people to deal with. Worldwide, the WHO says they're on the order of 500 million, and I'm sure that is an underestimate. These are people whose lives are significantly blighted by hearing problems, and they cannot all be treated with cochlear implants and things of this sort, which cost on the order of $50,000 apiece. But if these compounds could be used, we are talking about potential cures that would cost far, far less. So I'm hopeful that at least some of these will find clinical application. So I'd like to conclude. Thank you all for your gracious attention.
Thank you, Jim, for that inspiring lecture. We have time for questions. If you have a question, please raise your hand. We've got microphones in the aisles will find their way to you. So, there's a question here on the aisle.
Could you tell me to what extent artificial intelligence can be applied to these processes that you use for this discovery?
Sorry, I didn't get all of it. To what extent?
Artificial intelligence can be used in this process of discovery?
Well, that's a very interesting question. I'm sure every pharmaceutical company in the world is now trying to use AI. And the place that AI would be most valuable is when you know the structure of the target. When we did this experiment, we did not know what protein we were attacking, and the protein that we turned out to be attacking, it's called LATS, does not have an X-ray crystallographic structure. Some proteins, for whatever reason, crystallize well, and you can get their molecular structure. If you have that molecular structure, which is complicated, artificial intelligence is quite good at saying, here is the perfect molecule to fit in there and block it, or to fit in there and activate it. In our case, that wouldn't have worked because we don't have the structure. But I think that's where it's going to play out. And in fact, we have faculty members who are making use of this kind of approach to try to make highly effective, targeted molecules to affect particular biological processes.
Question right next to you.
Could you give us a little information about the instruments that were used to develop these frequency responses?
Say what you mean by frequency responses. In other words,
distribution of frequencies on that image.
I see. You mean just to make this kind of arrangement? Yeah. So what the ear does, as you've seen, is to break down complicated sounds into the constituent frequencies. And that's done by a process called Fourier analysis, after the engineer who first found this. But the idea is that any sound you hear, any, in fact, continuous signal, is always made up of sine waves. You can mix and match sine waves in different combinations to produce whatever you want. That's a surprising thing, at least to me. It's not at all obvious, but that's what Fourier showed. So any particular sound you make is going to be made up of sound waves. So let's take thank you all. So I'm from Texas originally, as Rick said, so I have a little bit of a drawl. The all has this nice stack of different frequencies, and there's a certain musicality to all. That's what you're seeing here. This is just like a note on a guitar or piano, and the harmonics that give it a certain musical quality. You don't see musical quality in the consonants. Gracious, you know, it's very noisy and hissy. So this is what our ears are figuring out. The way this is produced is that a machine takes the same information, it uses the same process, the Fourier analysis, to break down the sounds electronically, and then it simply makes a graph of how much energy there was at each particular frequency.
A question down here in the front row, and then back there.
Thank you, Jim. That was great. You started out by saying that one compound out of 140,000, but you ended up by talking about multiple compounds. Are there variations of this one compound?
So I subsequently mentioned that the Therapeutics Discovery Institute made 250 variants. So of the 16 million, or however many possible ones, I can see Lee, who's part of this enterprise, smirking because they know what a job this is. Of the millions of possible compounds, they tried to figure out which ones do we think might do the job. And this is a little bit like, say, cooking, with all due respect. You have a recipe that's so-so, and you try adding a little of this, a little of that, and it gets better or it gets worse. If it gets better, you add more of it, or you add something else. You know, you try to progress by making little tweaks, and many of those variants involved more than one tweak, adding a molecule or a constituent here, taking away one there. So they do their best to fill the possible space of recipes to find the best one of that group. And the best ones are extraordinary. I mean, the best molecules are 1 million times better at binding to their target than the normal molecule that binds there. So they're extremely effective, and very low doses will do the job.
And is that irrespective of the type of cell that you're trying to...
Yeah, so we don't really know that. So far, all the cells we've looked at behave in a fairly similar manner, and that makes sense in a way. Remember, our bodies have on the order of 20,000 different genes, and the particular protein that's involved here, there are actually 2 of them, are the same in every cell of our body. So once this compound or any of these compounds gets into the cell, we expect it will have pretty much the same effect wherever you happen to apply it.
Question back here in the back.
In the experiments you've shown, you've of course worked with mice as mammals. There are obviously non-mammalians that regenerate, and I'm wondering if you have seen a difference in the way these non-mammals do the regeneration, or if there's some aspect of what there is there to experiment with that might allow for certain other questions to be answered.
Right. So these same compounds work brilliantly in animals that do regenerate. So our lab does a lot of work with zebrafish, which is a model system for development and regeneration. And there also, the number of new cells being formed is cranked up appreciably by application of these compounds. But the issue you raise is one that's been a curiosity for, I presume at least decades, probably centuries. What is wrong with mammals? Why have mammals generically, and humans among them, lost so much regenerative capacity? So really young children, newborns, can regenerate fingers or parts of fingers. They can regenerate, some limited number of body organs, but soon after birth, that capacity disappears. Why does it disappear? And in terms of natural selection, is there some advantage? Is there some reason to lose that capacity? Or is this just, poorly designed software? And we don't know.
Question
couple of questions. One there and then over there.
When can we humans who are aging get the results and use the benefit that your investigation has led us to?
I don't know. I'm aging too, and I hope, I hope before too long. The key step for us now is moving from cells in culture and from small animals like mice to experimental systems that are closer to a human. And we hope, for example, possibly to use miniature pigs or the like. Those animals have a lot of physiology in common with human, some people in particular. And they're considered a very good model for many forms of human disease. So we hope within a few years to be able to step up to that level. And if that works, we would then be to the point of considering use in humans. There's one condition, very interestingly, that might be accelerated, and that is muscular dystrophy. Muscular dystrophy, Duchenne, Becker, is a terrible disease. It is uniformly fatal. It leads to severe muscle problems, but particularly to deterioration of the heart. So that might fall under the FDA's compassionate usage rules, which allow the use of compounds that haven't met all the criteria of testing in humans, simply because there's no alternative. So we'll see. We're not doing that work ourselves, but I'm hopeful that it will pan out, because if it did, it would be possible to bring these to use in humans within a few years. We'll see.
And right behind Corey.
As a follow-up to the last question, do you have a feel for how much of the progressive hearing loss, particularly in the aging, results from loss of hair cells as opposed to other disorders?
Yes, so that we do know. The great majority of all deafness lies at the level of hair cells. The estimates vary somewhat because there is clearly some pathology at the level of the ganglion cells. Those are the nerve cells that are connecting the hair cell to the brain. But in general, estimates are that perhaps 90% of all the loss is at the level of hair cells. So that's the target we need to regenerate. Rather remarkably, the nerve fibers typically do survive. That's why the cochlear implant works, because the nerve fibers are still there, they're just no longer getting any input. So if we could restore the hair cells, we think the thing would rewire and be effective.
Lulu has a question.
Thank you so much, Jim. I'm thinking as you are continuing to pursue the regeneration of hair cells, along the way, might you also, in understanding more of the mechanics of hearing, might you discover a new class of highly effective hearing aids?
While I'm up, you mean. There's a lot of effort being placed on things like that. The hearing aids have a limit. They have a problem. And that is, hearing aids amplify sound. But when you lose too many hair cells, even the amplified sound has nothing to detect it. So if you're losing, half of your hair cells or something, a hearing aid is extremely effective because it makes the sound somewhat louder. The surviving cells detect it. But imagine something like a person who's been subjected to some of the compounds I mentioned. He or she may have lost many or all of the hair cells. No amount of amplification is efficacious any longer. And that's when the cochlear implant or prosthesis is very valuable. It shocks the nerve fibers, which, as I mentioned, survive this kind of treatment. So I think the most exciting prospect is that the cochlear implants will get better, that it will be possible to do more processing to make better signals that go to the implant and make sound comprehension or speech comprehension more complete.
How far along are we in that?
So there are several different approaches, and there's a lot of advance being made. One idea is, instead of shocking the cells in the ear, the nerve cells electrically, is to use light, and to use light to trigger electrical responses in the cell, and there are technologies for doing that. The advantage is, When we shock nerve fibers by passing current into the ear, that current spreads and excites a lot of cells. And think about your body is all an electrical conductor, and things like your EKG can be measured in your fingertips because the signal goes everywhere. It's the same in the ear. If you shock a particular place, trying to excite a particular frequency, it will nonetheless spread to other cells, and you'll get a smear. With light, that doesn't happen. If you imagine a string of tiny LEDs along the length of the cochlea, each of them could hit just a few adjacent nerve fibers, and progress is being made on new prostheses, new implants of that sort.
A question right here.
Yeah, so the question is, how does the brain fit into the response? And remember, I said that we have this large number, about 15 or 16,000 hair cells. Each of them feeds into several nerve fibers that go into the brain. So at the other end of those nerve fibers, the brain has a series of nerve fibers, each of which represents a specific frequency. Now, the brain needs to use that information to figure out what sound is being heard. And so the brain has to learn that information, and in particular, when we learn speech, our brain learns to associate this frequency, that frequency, and the other frequency, say with a specific vowel. And that's what you see again here. There are subtle differences. It's not obvious here, but you can see, for example, in this thank, you can see the spacing here between these frequencies is coarser, broader than the spacing, say, here, or in some of the other cases. So those are called phonemes, or speech sounds, and the subtle spacing differences and the like, how far apart the tones are, is what the brain learns to interpret as a specific speech sound.
So to follow up on that, if you were to replace hearing in somebody who had been hearing but had lost their hearing, and you replaced their hair cells, so the signal was now being transmitted, has the brain retained the information in how to decode, or is it going to have to learn it again?
So that's an essential part of the way that the prosthesis works, that it is extremely effective in people who are so-called post-lingually deaf. These are people who have had normal hearing, normal speech recognition, and so on, and who have lost their hearing for one of the reasons that I mentioned. People of this sort need very little training to make use of the prostheses. And there was an incident along these lines when I was at University of California, San Francisco. This was a new technology. And there was a woman who was flown in from Ireland because she had hearing loss, but there was the possibility of replacing things and putting in a prosthesis. And she was fitted surgically. She came out of the anesthesia, and she couldn't understand anything. It really didn't work until somebody in her family came in and said, "Oh, in Begorra, how are you today?" She said, "Oh, I'm fine." So, immediately she understood perfectly. It just, she was trained on a particular accent.
A question in the back row.
The person under the tripod has been trying desperately to be recognized.
He's moving the camera.
Is that what it is?
Just to take your really quite brilliant work and simplify it, at least the way I'm understanding it, are basically you're looking for how to control neurogrowth factor and transcription factors in specific cells in the body.
That's correct. And where that's concerned, I want to make another point about this. Turning on all the cells of the body, saying let everything that wants to reproduce, could be dangerous, right? That could lead to cancer, it could lead to other bad things. But there are two mitigating circumstances. One is we just need one round of cell division. We're not talking about something people would take daily for life. What would take one dose, for 3 days is what it takes. That would be enough to cause the one round of replication in the cells, and that's all we need. If we could double the number of supporting cells, we think we could make sufficient number of new hair cells. The second thing is we would like to direct the compound to specific places. So for example, in the ear, it's possible to do surgery or to penetrate the eardrum and put a compound just in the ear so it doesn't go elsewhere in the body. In the eye, that's almost trivial because one can inject into the eye, as one does right now with therapies for various retinal conditions. In the heart, there's a pericardium, a nice sac around the heart. It would be possible to put a drug there and to affect largely the heart, but not other organs. So we think this localization of the therapy will also be quite important.
Terrific. I think we're at time. So once again, I'd like to thank Jim very much for an enlightening talk. Thank you very much. It was spectacular.